This is the inter chain. Okay? But sometimes, for example, you have the N to C, it's fold on itself, NC, and you have a hydrogen bond here. Because only you have the one polypeptide, this is known as a intra-chain. Do you understand? Inter-chain, intra-chain actually. But about the collagen, we have a inter. For example, you have a 3 alpha chain, and between chain you have a interchain hydrogen bond. About the some specific characteristic of a structure, at first you should know that we have a wide distribution. This slide is for more information. You know, I put intentionally because I want to represent the wide distribution and the different form of the collagen. Sometimes it forms a gel. For example, in a vitreous humor. Do you know what's a vitreous humor? Okay, vitreous humor. And also, for example, they present as a, for example, some parallel fiber, they provide a strength, and sometimes they present in an eye, in the bone, in different, for example, in a tendon. Without microphone, I cannot speak. Okay.
Not important.
No, for a final exam is not important. Because I want to present a different form of the collagen. Okay? I wait for a microphone. A different part of the body. Again, this slide is more informative. It's for more information because I want to represent the diverse function of the collagen. What about this slide? This is a general characteristic of the structure of the collagen. At first, you should know that which amino acid frequently found in a structure of the collagen. As I mentioned, because it is a 3 alpha chain, they wound around one another, okay, it's better to have some small amino acid. So, at first, glycine is very suitable. And also proline, because inverted king and angle is a suitable amino acid. But we have a repeating sequence in the structure of the alpha chain. You know, if you evaluate the structure of alpha chain, you have a repeating sequence. Glycine X, Y, glycine X, Y, okay? So glycine is fixed. What is the X actually? X frequently is proline, and about the Y is a hydroxyproline and hydroxylysine. I think in your high school you're familiar with the post-translational modification. You know, it means that after translation you add some group like a hydroxyl, okay? So you have another type of the amino acid, I mean after translation, they take, for example, hydroxy group here, okay? This is known as a post-translational modification. And also you should know that collagen is a glycoprotein, okay? Some chains bind to the glucose. Some of them, they bind to the galactose. So we can say that collagen is a glycoprotein amino acid. But About the biosynthesis of the collagen, many cells of the body can synthesize the collagen, like a fibroblast in a connective tissue, like an osteoblast in a bone, chondroblast in a cartilage. This is a general pathway of the collagen. If you want, you can review, but I never asked you about the details of the biosynthesis. But between this long biosynthesis pathway, for example, at first, you have a translation of the chain. Add a hydroxyl, add a glucose and galactose, and they wound around one another to finally reach to the last level of structure of collagen. But about this pathway, you have a one specific reaction. And because as a result of deficiency of this reaction, we have a one disease, scurvy. Have you ever heard this name? Scurvy, it's bleeding gum. As a result of deficiency of vitamin C in a sailor, okay? So, one of the important reactions in the metabolism of the collagen is the hydroxylation of the proline and lysine. Inshallah, on Tuesday I will describe about the enzyme. Whenever you have an ACE yani enzyme, hydroxylase, kinase, urease, ACE yani enzyme, okay? During the biosynthetic pathway of the collagen, as I mentioned, you have a prolyl hydroxylase. These are enzymes, lysyl hydroxylase. For example, here, for example, this is proline amino acid. After hydroxylation, lation is a French word, you know, lation yani process, okay? Hydroxylase yani enzyme, lation yani process, okay? Hydroxylation, the name of the enzyme is hydroxylase, okay? And this is the name of the substrate. Prolyl hydroxylase yani you add the hydroxyl to prolyl. Lysyl hydroxylase yani you add the hydroxyl to lysyl. Okay? What about the necessity of the activity of the hydroxylase? I'm talking about this enzyme. You require ascorbate. What is ascorbic acid? Have you ever heard this name? Vitamin C. Excellent. This is the scientific name of vitamin C. So, for adding hydroxy to proline, you require ascorbic acid or vitamin C. So, what happens whenever vitamin C is deficient? You cannot add a hydroxyl. You know? What is the reason for adding hydroxyl? I want to explain it. This is a string of your collagen. For example, I draw 2 chains, okay? But absolutely you have a 3 alpha chain, okay? Why our nature, our body, added hydroxyl? For example, here you have a lysine, here you have a proline, okay? Why? What is the reason for adding hydroxyl here? What do you think? Why Allah add more hydroxyl?
For sterilization?
For what is the end reason?
Digested to the water, like more.
Oh, no. Water, no.
No, the solution, not.
For connection with.
For connection with water, yeah. Solution.
Please recall that. Yes.
I think it's used for the synthesis of collagen. So you need this group because it's the functional group that is required in order to do a reaction.
Wait. Recall the bond between alpha chain. As I mentioned, what is the bond between alpha chain of the collagen? Hydrogen bond. Am I right? You have a hydrogen bond here. Am I right? For stabilization of your collagen. So what is the reason for adding hydroxyl?
In general, hydrogen is not.
Excellent. To provide the hydrogen for making hydrogen bond. You understand? The reason of why we add OH to proline, why we add OH to lysine for stabilizing and actually for facilitating the formation of the hydrogen bond. So what?
Professor, can it be considered as hydrophobic interaction?
Not hydrophobic. OH is actually polar group actually. OH can participate in a hydrogen bond. Okay? As I mentioned, because you have an H bond here, for facilitation of the H bond, you added more OH. But please imagine, whenever hydroxylation is معيوب, what happened? Is defective. What happened? Collagen is معيوب. Okay? So, as a result, you have a scurvy. Okay? Whenever collagen is defective, معيوب. Okay? You have a one disorder known as a scurvy. So everywhere of the body, like blood vessels, you have some bruise. Okay? This is bruise. Because the blood vessels are unstable. You have a bleeding. And for example, in a gum, we have a collagen. You have a bleeding gum. Okay? So these are examples of the symptoms of the deficiency of the vitamin C. And now you know that why deficiency of vitamin C causes scurvy. Because deficiency of vitamin C can destroy this reaction. So as a result, you have the abnormal collagen, like a gum, like a blood vessels. So you have some problem regarding to the structure of the collagen. About this slide, just know that about the name. Inshallah, in future you will be familiar with the details. You know, we have some disorder in addition to scurvy that are related to the collagen. So, what is the sample question? Maybe I ask you. In scurvy, the biosynthesis of which protein is defective? Hemoglobin, myoglobin, collagen, elastin. Collagen. Another question. In Ehlers-Danlos syndrome and in osteogenesis imperfecta, or other name is brittle bone disease, what is the name of the protein that is defective? Collagen. Inshallah, in the next semester you will be familiar with the different type because, for example, osteogenesis imperfecta can classify as a mild to lethal. Okay? It takes place during the infancy. As you can see here, the bone contains collagen. So, during the pregnancy, the bones are very fragile. They can break very easily. Okay? So it is osteogenesis imperfecta. And about the Ehlers-Danlos, especially the collagen under the skin, they have an abnormal stretchy skin. Okay? But the type is not important. Just know that Ehlers-Danlos and osteogenesis imperfecta are examples of the disorder of the collagen. About another protein, this is elastin actually. Have you ever heard elasticity? Like a rubber. Where we have elasticity? Lungs. Okay? After taking deep breath, you increase the size of the lung, okay? And after exhalation, you decrease the size of the lung. So in the lung, you have an elasticity protein, elastin. It acts as a rubber, okay? So it's composed of actually connective tissue is full of elastin. As I mentioned, it acts and functions as a rubber. And as I mentioned, it can stretch and relax, okay? Stretch and relax for several times. What is important about the elastin, especially for your final exam? At first, you should know that this is a insoluble protein. Okay, for example, hemoglobin is soluble, but elastin, because it's structural protein, is insoluble. Another point about the name of the precursor is tropoelastin. And in some parts of the body, in an extracellular space, we have this protein. But I want to direct your attention to the fibrillin. These are fibrillin. You know, they protect the structure of the elastin. They act as a scaffold. They protect, they have a protection, okay? So, sometimes elastin is normal, fibrillin is بايو, okay? So, you should know that we have one supporter, okay? And the name is fibrillin, okay? To protect the structure of the elastin. What about the structure? What is important to know? It's full of proline and lysine. Actually, a little hydroxyproline and little hydroxylysine is present, but we have a specific residue of the amino acid, and this is lysine. What is it? I know that this is one of the basic amino acid, lysine. We know what is lysine. But after oxidation of lysine, it converts to the aldehyde form, and the name now it is lysine. If I ask you, in the structure of which protein lysine is present, what is the name of the protein? Elastin, actually. And whenever we say that, for example, we have a stretchy form, okay, you can have elasticity. Which structure provided this stretch actually form? You should know that 3 lysine bind to the 1 lysine, okay? They form this structure, and this structure known as a desmosine. Now you can stretch from any direction, you know. 3 lysine and 1 lysine amino acid, they bond together and they form a ring. And from each direction of the ring, you can stretch your molecule, okay? So you have this structure, you can stretch. Do you understand? You can stretch this molecule. And why we say that elastin is a stretchy molecule? Because of the presence of desmosine. So, another question, whenever I ask you desmosine, what is desmosine at first? And you should know that what is the component of desmosine? This is the 1 unaltered, unchanged amino acid lysine with the 3. After oxidation, you have a lysine. 3 lysine amino acid, they form a desmosine. They present, as I mentioned, in a connective tissue, and they provide elasticity. About another disorder that is related to the fibrous protein is a Marfan syndrome. Sometimes I ask you, the name of the protein is defective in a Marfan syndrome. What is the name of the protein? Fibrillin, excellent. You should know that in a Marfan syndrome, elastin is normal, okay? But a scaffold is abnormal. I mean, fibrillin is abnormal. But about the details of this disorder, it's a mutation, there is no need to memorize. Just know that Marfan is as a result of deficiency of which protein? Fibrillin, okay? About the degradation, also this is for more information. If you want to take in more information about the occurrence of the emphysema, you can review this slide. What about this slide? We talk about, but I never ask you about this slide because I know that you are familiar. Collagen may different genetic collagen type 1, type 2, type 3, okay? But only one type of the elastin is present. If you remember, you have a repeating structure. There is no repeating structure in elastin. Carbohydrate present in elastin is protein. Yani collagen is glycoprotein. Elastin is not glycoprotein. Desmosine is present, and also, for example, they present in an extension of the peptide during biosynthesis and no extension peptide. This is regarding to the biosynthetic pathway. As I mentioned, this is an overview of the comparison between elastin and collagen. What about the final exam? I never ask you about this slide, okay? Because I focused on the main important in the previous slide. About the globular protein. About the globular protein, we have two different globular proteins, hemoglobin and myoglobin. At first, you should know that hemoprotein, what does it mean, hemoprotein?
blood protein.
Protein found in the blood.
No,
The
no,
plasma of the blood.
no,
The red blood cell.
no. Hemoprotein is different from hemoglobin, do not make a mistake. Protein, for example, this is a large protein. Whenever at the middle, for example, you have one group known as a heme, generally we say that this is a hemoprotein. We have another example, different example of the hemoprotein. One of them is hemoglobin. Okay. Another one is cytochrome. Maybe I ask you about this name. Catalase, hemoglobin, and myoglobin. So the first important point of this slide is the name of the, for example, maybe I ask you, all of the following considered as a hemoprotein. Cytochrome, I don't know, catalase, hemoglobin, I don't know, elastin. Elastin is not hemoprotein, okay? So at first you should know that. What is the example of the hemoprotein? As I mentioned, this protein, they contain a heme group. And heme group is a prosthetic group. Prosthetic group is that some complementary molecule, they bind to the protein to complete the function of the protein. Okay? For example, inshallah, during the vitamin session, Dr. Khalili will describe. We have some vitamin considered as a prosthetic group. For example, vitamin B1 added to some enzyme to accelerate and actually complete the function of the sum of the enzyme. So prosthetic group means some group that tightly, with a strong bond, attached to the another part. For example, this part is protein, this is a prosthetic molecule. As I mentioned, we have a different type of the hemoprotein like a cytochrome. Cytochrome, have you ever heard this name? Are you familiar with the electron transfer chain
Yes.
in the mitochondria?
Yes.
Okay, some part of the electron transfer chain is cytochrome.
So there's a curved cycle.
Not curved cycle, in the electron transfer chain of the membrane of the mitochondria. Yes, ETC, electron transfer chain, actually. And they actually detoxify some drugs, actually, they have a different role. Catalase is one of the enzymes that is important for the antioxidant defense, and hemoglobin and myoglobin are the most important heme protein in the body, and in the next slide, I will discuss about this actually protein. Before that, I want to review the structure of the heme. What is the structure of the heme? In this actually picture, you can observe the overall structure of the heme. At first, I want to direct your attention to the form of the iron. What is it?
Ferrous.
What is it? Fe3+.
What's the value?
Ferric. Okay? You should know that if you change the ferrous to the ferric, it can cause a death. Because Fe3+ cannot bind to the oxygen. I'm talking about the hemoglobin. At the middle of the hemoglobin, you have a heme. At the middle of the heme, you have an iron atom. Okay? And the type of the iron is very important. Absolutely, it should present as a ferrous atom. Because whenever it binds to the Fe3+, hemoglobin changes to the met hemoglobin. And met hemoglobin cannot take oxygen, so you cannot take breath. Okay? So the form of this iron is very, very important. And as I mentioned, it's located at the middle of the structure of the hem. This is, for your level, there is no name, but just know that. About the name of the ring is a protoporphyrin 9, and this is a porphyrin ring, okay? Inshallah, during the metabolism of the nitrogen molecule, you will be familiar. What is important here? You should know that we have one specific amino acid in the structure of the hemoglobin and myoglobin. This is a histidine, as I mentioned. And as I mentioned, it has a very important role, and most of the time considered as a bufferic amino acid. It can bind to the, and also the heme group can bind to the oxygen. As you know, the role of the hemoglobin is a transpiration of the oxygen from the lung to the